I'm going to walk through analysis of a fake practice mutation just to show you guys all the steps. So these are the 4 that you guys are going to work on, but I just made up an extra fake one for the purposes of this little video. All right, so at this point, you should already have imported the E. coli genome into your Benchling. So that's what I'm looking at here. So this is the entire, 4.5 million, you can see down here in the left, nucleotide sequence of the genome. These little arrows are the genes, right? So gene number 00010 doesn't have a name, but it has a potential function, right? Bifunctional aspartate kinase, almost zero dehydrogenase 1. Sure. They're annotated from start codon to stop codon. So this flat end of the box is the start, and then it goes towards, this is a long gene, the arrowed end, which you can see here. This is the stop codon. You see how close these are together? This is what operons look like in E. coli, right? These coding regions are only one nucleotide apart. There's no room to put a promoter here. These are going to be made as one big long RNA, which is cool. Operons are cool. All right, so let's get started. We're looking for position number 12301. It's not really a good way to do this other than to just scroll around. If you need to go really far, you can click in the middle over here to jump long distances and kind of find your way around. But of course, my fake one is close to the top at only 12,000, so I don't need to scroll for all eternity here. 12301. Yeah, 12301. All right, so you can start looking around. 12301. Okay, so I highlighted a base to see if I had the right one. Down here at the bottom, you can see start and end numbers are 12301. So I've got the correct base. I'm going to mark it by creating an annotation. So up here on the right, create annotation. I'm going to name it 12301. Scroll down, hit save. The little arrow that it made is really tiny, but it's convenient because you can now click this and find it. And it means that if I use my little search bar right here, I could do 12301 and hop right back to this should I scroll away to look at something else. All right, so I have found it. The reference should be a G, which it is indeed a G in the reference sequence that I am looking at. So I should be in the right spot. All right, it was changed to a T. We'll figure out what that means in a minute. These AD numbers, which were pulled from the other one, are the number of reads with the reference and the number of reads with the alternate. So to find percent of reads with mutation, you would just do 2. Well, it's better to do it this way. 2 divided by 2 plus 222, right? So the total number of reads is the two numbers combined to find the percent that have the mutation. Oh, wait. Y'all don't listen to me. I did that upside down. Mutation divided by total. I even wrote it for you right here in the formula. Follow the formula. And then look, it's going to be smart and be like, do you want to do all of them? You should, but I'm going to click no. All right. And it's looking like 99.1%, which fits with the numbers we're seeing, right? 222 reads with the alternate, only 2 with the reference. All right, moving on along. Is the mutation in a gene/coding region? Well, it is because it's in this coding region, right? So let's say yes. Yes. If yes, did it change the protein? All right, we'll get to that in a second. Gene name and or number. Oops, wrong way. 0070. This gene also has a name. DNA K. E. coli genes are always usually 4 letters long, right? That you capitalize it if you're talking about a protein and you don't italicize it. If you're talking about a gene, you do lowercase and italics. Annotated function, molecular chaperone. I spelled that wrong, but you know. Okay, so let's go back to did we change the proteins so you can understand how to do that part. Wrong way again, sorry. All right, we are in a coding region, which means this is translated, right? So our codons are ATG, always our start codon, GTA, AAA, right? It would take you forever to stare at that and figure out what this one is. We're not going to do that. We have a computer. We're going to do create, translation. This gene is going in the forward direction, right? Because the arrow is pointing down. The arrow is not on this end. So we do create translation forward. Name it the gene number 00070. Scroll down. Hit save. It will translate it for you. Isn't that beautiful? You can click this to make this go away. All right, there's your codons. Glorious. All right, so now we can come over here and click on this and see where it is in our codon, right? So it's in this E glutamine codon. That is GAA. So if we're smart, we're going to have another tab open, right, with our handy dandy codon chart, which we have been using this one in class, but you're welcome to use a different one if you like it better. GAA. Glutamine. Glutamic acid. Sorry, I'm confusing myself. I hate the 3-letter codes. I don't have them well memorized. Glutamic acid. Art. What did we change it to? Scroll back over here. It started as a G. Now it's a T. Right? You're in our alternate base. So that means GAA became TAA. Back to our codon chart. Sorry, wrong tab. T becomes U in our RNA. UAA is a stop codon. So what does that mean? It means we went from E to stop. So we've got a nonsense mutation, and it's an early stop at amino acid 47. So I'm going to put yes. Early stop at AA 47, or the proper way to do that would be E47. And then just because it's interesting to note how far into the protein we stopped. Like, is 47 the end? Is 47 the beginning? Like, how long is this thing? Oh, it's long. Oh, it's real long. It's 638. 638 days. So very early stuff. All right, that's cool, right? So now you can look at this and be like, hey, this is a really early stop codon in this gene or in this protein, right? So very most likely this protein is effectively non-functional, right? We lost the vast majority of this protein at the translational level because we have this early stop codon here. And so then you're going to think about, right? All right, what does this thing do? We think it's a molecular chaperone. Go look up what that is. What might it mean to lose a protein like that? And how does that relate to the resistance that you've evolved? Get to those later steps later, but the process of filling out this chart for each mutation of interest, that's what it's going to look like.